Hey, you. Ever been completely absorbed in learning something new and wondered what's actually going on inside your head? How does your brain take something unfamiliar, whether it's, a tricky new skill, the lyric to that earworm of a song, or even just remembering a new route and make it stick?
That's a fantastic question. It really gets to the heart of neuroscience, doesn't it?
Yeah.
And the fundamental answer is that learning physically changes your brain. It actually rewires itself.
Rewires.
Yeah, by reorganizing the trillions, and I mean trillions, of connections between your brain cells, your neurons.
Okay, let's unpack this a bit. So we have these billions of neurons, right?
Yeah.
And they talk to each other across these tiny gaps called synapses.
Exactly. Synapse.
Like if the neurons are roads, the synapses are the traffic lights, controlling the flow.
That's a great analogy, yeah. And it's at those traffic lights, the synapses, where the real action of learning happens.
Okay.
When you learn something, it's a very dynamic process. Some of these synaptic connections become stronger.
Like widening the road.
Precisely. Making it easier for traffic or information to flow in the future. And other connections weaken.
Like paths getting overgrown if you don't use them.
You got it. This strengthening and weakening process, we call it synaptic plasticity. That's the core mechanism your brain uses to encode new experiences.
Right, plasticity. Okay, so that leads us to this big, long-standing mystery neuroscientists have been wrestling with. What are the specific rules that decide which connections get stronger and which get weaker during learning?
Yes, the rules of the game, so to speak.
And why should our listeners care about these rules?
Well, understanding these rules is it's like unlocking the secret instruction manual for how your brain actually acquires knowledge and skills, everything you know.
Wow, okay.
And that's exactly why this groundbreaking research just came out of UC San Diego is so exciting.
Ah, okay, the new study.
Yes. William Jake Wright, Nathan Hedrick, and Takaki Komiyama. They published this fascinating study.
Where was it published?
In Science, just recently, April 17th, 2025. And it sheds a whole new light on these exact rules of synaptic plasticity.
And this wasn't just a quick look, right? I saw it had significant funding.
Oh, absolutely. It was a multi-year project heavily backed by the National Institutes of Health, the NIH.
That signals it's pretty important stuff.
Definitely. It underscores the importance and, frankly, the rigor behind their findings.
And here's where it gets really interesting for everyone listening. This isn't just about, satisfying scientific curiosity.
Not at all.
Understanding these fundamental brain mechanisms has some potentially huge implications down the line, doesn't it?
It really does. We're talking about the possibility of gaining new insights into treating brain disorders.
Okay, like what?
Well, we'll get into specifics, but think Alzheimer's, PTSD, addiction, and even things like inspiring the next generation of artificial intelligence.
AI, too. Wow. Okay, so the implications are broad.
Very broad. But maybe let's dive into how they actually arrived at these new insights first, because the methods were pretty ingenious.
Yeah, let's do that. How did they see this happening?
They used some really cutting-edge brain visualization techniques, most notably something called two-photon imaging.
Two-photon imaging. Okay, That sounds incredibly advanced. What does that actually let them do? What's the advantage?
Well, it's amazing, really. It lets us see much deeper into living brain tissue than traditional microscopy.
Okay.
And crucially, we can observe fluorescently labeled proteins involved in synaptic activity without causing significant damage to the tissue.
Ah, so they can watch it happen in a living, learning brain.
Exactly. It allows researchers to study the dynamic changes happening at these tiny synaptic connections in real time as learning is occurring.
So it's like having this superpowered but gentle microscope watching the brain learn, literally synapse by synapse.
That's a perfect way to put it. Microscopic level detail as it happens.
Okay, so with this incredible view, what did they find that really shook things up? What was the big surprise?
The key finding directly challenges a long-held assumption in the field.
Which was?
The prevailing idea, sort of the standard model, was that individual neurons largely followed a uniform, single set of rules for how their synapses changed during learning. One rule book per neuron, basically.
Okay. Made sense, I guess. Simpler.
Seemed simpler, yeah. But their detailed observations revealed something far more intricate. Individual neurons actually appear to follow multiple rules simultaneously.
Wait, multiple rules?
Yeah.
Within just one single neuron?
That's the kicker, yes.
Wow. Okay, that's really surprising. So it's not a one-size-fits-all approach even within the same nerve cell?
Not at all. They found that synapses located on different branches of the neuron, these branches are called dendrites.
The input branches.
Right. Synapses on different dendrites of the same neuron were operating under different principles of plasticity, different rules for different neighborhoods, you could say.
That suggests a much more sophisticated and nuanced learning process than we thought, happening right inside individual brain cells.
Not more sophisticated, yes.
So, okay, if we have these neurons and different parts of them are following different rules, that brings up this other big question, right? This credit assignment problem.
Ah, yes, the credit assignment problem. That's a critical concept here.
Explain that a bit. How does it relate?
So the credit assignment problem in neuroscience basically asks, how do individual synapses, each one only having access to very local information like what's happening right around it, how do they know how to adjust themselves to contribute to a complex, brain-wide learned behavior?
Right. Like, how does the tiny synapse know it helped me learn to ride a bike, not just that some signal passed through?
Exactly. It's like trying to build a huge, complex machine where each tiny screw only knows about the piece it's directly touching. How does the overall design get implemented correctly? How does the credit for success or blame for failure get assigned back to the right tiny components?
Okay, I see. So how does this new finding the multiple rules thing help with that problem?
Well, it offers a really intriguing potential answer. The fact that different parts of the neuron, different dendritic compartments, seem to be governed by different rules suggests something quite profound.
Which is?
That perhaps the neuron itself is performing multiple distinct computations in parallel.
Whoa.
Different dendritic compartments might be responsible for processing different aspects of the incoming information or contributing to learning in different ways using different rules.
So the neuron isn't just a simple relay switch. It's like a mini computer with specialized processors.
If that's a way to think about it, yeah. A mini processing unit with different tools or rules for different jobs, all within one cell.
That is genuinely mind-blowing. I read the researchers themselves were pretty taken aback by this level of complexity.
They were. They really emphasized that the ability to simultaneously visualize both the inputs coming into the neuron and the resulting changes at the synapses all in real time, that was absolutely crucial.
Without that two-photon imaging.
They wouldn't have seen it. They could literally watch these different rules playing out in different parts of the same neuron as the mouse was learning. It was visible.
Incredible. And they had some insightful things to say about what this discovery means, right? Any key quotes that stood out?
Yes, definitely. Dr. Wright, one of the lead authors, he highlighted that their research gives us a much clearer picture of this fundamental process, how synapses actually change when we learn.
Okay, a clearer understanding.
But he also pointed out the potential health implications. Understanding this normal process in such detail could be really important for diseases where synaptic function goes wrong.
Right. You mentioned Alzheimer's earlier.
Exactly. Imagine if we could figure out which specific plasticity rule is malfunctioning in a condition like Alzheimer's or maybe certain forms of autism.
You could potentially develop therapies to target just that rule.
That's the hope. Much more precise treatments that correct the specific problem at the synaptic level.
That would be huge. And Professor Komiyama, the senior author, he really focused on how this changes our view of that credit assignment problem, didn't he?
He did. Professor Komiyama explained that this finding fundamentally shifts our perspective. The idea that single neurons can perform these distinct computations in parallel in different subcellular compartments.
Yeah.
It suggests a level of computational power and sophistication within just one brain cell that, honestly, we hadn't fully appreciated before.
So for us, for the listener, what does that mean?
It means your brain is even more adaptable, more intricate, and frankly, more amazing than you probably ever imagined.
Okay, my brain feels a bit more amazing now. So if our brains are doing this incredibly complex dance with multiple rules inside single cells, what about artificial intelligence? You mentioned that connection.
Yes, that's a really interesting angle.
Because a lot of AI development tries to mimic the brain, right?
It does, often quite loosely, but the inspiration is there. Now, current AI neural networks typically operate using just one common set of rules for adjusting their connections, their artificial synapses.
One rule book for the whole network, or maybe per layer.
Right. This research suggests a potentially powerful new direction. What if we designed AI systems where individual artificial units, like artificial neurons, could also utilize multiple plasticity rules simultaneously?
Mimicking the biology more closely.
Exactly. It might allow us to create AI that learns in a more brain-like way, perhaps much more efficiently or more robustly, especially when dealing with complex, changing environments.
Huh. Current AI can be a bit brittle sometimes, struggles with new situations.
It can be. Mimicking the brain's ability to use different learning strategies in parallel, even within one unit, might help overcome some of those limitations. It could lead to more adaptable AI.
Fascinating. So instead of one master algorithm for learning in the AI, different parts of the artificial neuron could follow different learning procedures. Okay, let's circle back to the potential for treating brain disorders. You mentioned a few earlier.
Yes, this is really where the hope lies for tangible benefits.
How does knowing about multiple plasticity rules help with something like, say, addiction or PTSD?
Well, think about it. If we can get a really detailed map of how the brain normally encodes information using these multiple rules at the synapse level.
The baseline, the healthy function.
Right. Then we can start to pinpoint what might be going wrong in conditions like addiction, where learning pathways become pathologically strong, or PTSD, where fear memories are incredibly persistent, or Alzheimer's, where synaptic connections are lost, or even neurodevelopmental disorders like autism, where synaptic development might follow a different trajectory.
So maybe in addiction, one type of plasticity rule is running overdrive, or in Alzheimer's, certain rules just aren't working anymore.
Potentially, yes. By understanding the normal rulebook in detail, including all the different rules, we can identify which specific rules are disrupted or misapplied in these conditions. And that knowledge is the first step towards developing targeted therapies to correct those specific synaptic malfunctions.
It moves us beyond just general ideas about synaptic problems to potentially identifying very specific mechanisms to target.
Precisely. More targeted, hopefully more effective treatments.
So this study is obviously a huge step, but it's not the end of the story, is it? What are the researchers looking at next? Where does this go from here?
Oh, absolutely not the final word. If anything, this discovery has just blown open a whole new area of questions.
Like what?
Well, your researchers are now digging into how. How exactly are neurons able to manage and utilize these multiple rules at the same time? What are the molecular mechanisms inside the neuron that allow different rules to operate in different dendritic compartments?
And also why, presumably? What's the advantage?
Exactly. What are the specific benefits of having this complexity? Why did the brain evolve this capability? Does it allow for faster learning, more robust memory, better adaptability? These are the big questions they're tackling now.
So it's very much an ongoing active area of research?
Very much so. This study is a launch pad.
Okay. And for anyone listening who really wants to get into the weeds, read the original paper. Can you give us those publication details again?
Sure thing. The study is titled, Distinct Synaptic Plasticity Rules Operate Across Dendritic Compartments in Vivo During Learning. The authors are William J. Wright, Nathan G. Hedrick, and Takaki Komiyama.
And it was in Science.
Yes, Science Journal, published April 17th, 2025. And if you want to find it online easily, the DOI, the Digital Object Identifier, is 10.1126science.ads 4706.
Great. And you mentioned the funding sources earlier too, right? NIH.
Yeah, it's worth noting the breadth of support. Again, NIH, the National Science Foundation, and SF, the Simons Foundation, G. Schmidt AI and Science Fellowship. It really highlights that this kind of significant fundamental research often requires collaboration and substantial backing.
Absolutely. Okay, so as we wrap up this deep dive, what's the main thing you want our listeners to take away from all this?
I think the key takeaway is that learning deep down at the cellular level in your brain isn't some simple, uniform process. It's incredibly sophisticated.
Not one size fits all.
Definitely not. Your brain, right at its most fundamental connections, those synapses, uses a diverse toolkit, a whole set of different rules to encode new information.
Yeah.
Even within a single brain cell, it's far more complex and capable than we often give it credit for.
And for you, the listener, especially if you're interested in learning and gaining knowledge quickly, but also thoroughly, this research just really highlights the incredible machinery you have upstairs. It underscores the amazing complexity and efficiency of your brain's learning mechanisms. It's a testament to the adaptability that lets you absorb and integrate new stuff constantly.
Exactly. And, it leaves us with a really provocative thought to mull over.
Well, one.
If individual neurons can juggle multiple learning rules at the same time, what other hidden complexities might be at play in how we acquire, store, and use knowledge? We're just scratching the surface here.
Right. And what might this eventually imply about, I don't know, optimizing our own learning? Could we potentially leverage this understanding in ways we can't even imagine yet?
It's a fascinating possibility, isn't it? What untapped potential lies in understanding these intricate rules? It's something to think about. This research has definitely opened up a whole new landscape of questions and possibilities.